found this finding to be fascinating. It really wasn't something that we had on our radar, but Crystal Mackel at Stanford, working alongside David Miklos, running the clinical program there, found that CD58, also called LFA3, it's known to have mutated or been lost in about a fifth of patients with diffuse large B-cell lymphoma. But what they found is that patients who did not have CD58 by immunohistochemistry, that these patients had extremely poor outcomes, progression-free survival outcomes, compared to patients who did not have this loss of CD58 by immunohistochemistry or by deep sequencing they had done on circulating tumor DNA. And they took it a step further. They actually manufactured a CAR that signaled through CD2, which is the receptor, or excuse me, the ligand for CD58. So CD2 is the ligand. And so they induced CD2 within the CAR, the signaling for it, and as well as CD2 presentation in trans on these CARs. And they found that they could overcome CD58 loss in target cells in vitro and in vivo. And I think this is a really nice example of using, bedside derived material on CAR-treated patients, finding a new finding that is associated with lack of response and durable response, and then actually creating a CAR T cell that can potentially overcome that mechanism. So I really think this is an important finding, but also an important sort of roadmap for how the field can best utilize patient samples and come up with treatments that might be better for patients ultimately.